I'll talk a little bit more in depth later about some of the specifics of the projects that my lab's working on, but I largely wanted to focus on one of the major areas that we're studying, which is the evolution of dosage compensation. And I'm here in the School of Life Sciences. I'm Melissa Wilson-Sayres. Sometimes we forget to introduce ourselves. Affiliated with I'm a core faculty of the Center for Evolution and Medicine and affiliated with the Biodesign Institute here. So a note about language is that unfortunately the language we use for sex and gender is the same. And so when I'm talking about males and females, especially in humans, I mean specifically individuals that are passing on their DNA via an X and Y chromosome in sperm as males, and individuals who are passing on their DNA via 2 X chromosomes in eggs. So we know there's a spectrum of intersex individuals and also a spectrum of gender identity. And here I'm really talking about separate sexes. Great. Okay, so we're all here because we're interested in evolutionary genomics of sex, and that can be sex determination, sexual conflict. For me in particular, I'm interested in studying sex chromosome evolution, and largely I've studied it in humans. And so here's a picture of a karyotype of humans with the GIMSA staining and the X and Y chromosomes there. And so I'll focus a little more because we've had a lot of background on some different aspects, but in mammals and humans specifically, we think that the X and Y chromosome evolved from an autosomal pair about 160 million years ago. So when we were first studying sex chromosomes, we thought that the X and Y were shared across all mammals, and so we thought they were about 200 or 300 million years old, but now we know the monotremes have an independently derived sex determination system. So our X and Y of eutherian mammals and that of the marsupials is actually shared. So about 160 million years ago, we think, because it's shared across marsupials and eutherians, a pair of autosomes started becoming differentiated into sex chromosomes. So unlike the very young systems that Thiele and Ashman discussed earlier, we have really old sex chromosomes and pretty differentiated sex chromosomes. So the X chromosome in humans has about 155 million sites and 1,100 genes, give or take. The Y chromosome has about 60 million sites and 27 unique genes. Some of these have multiple copies, so we can have as many as 76 copies of some of these genes, but about 27 unique protein coding units on them. And so if we just look at those numbers today, and you believe me that 160 million years ago they were largely homologous, how did they become so different? And so we have to try to understand how they became so different before we can think about dosage compensation. So here's our ancestral autosomal pair. And we'll make it into a cartoon here so we can just think about different loci. And each one of those loci can recombine. So they're homologous, they can swap DNA, you can fix errors that happen in between the 2 homologous chromosomes on our ancestral autosomal pair. And probably most talks here will cite the Charlesworths, and it's unfortunate we couldn't have them here today, but we have some hypotheses for why we can get sex chromosomes. And so earlier we heard about female heterogametic systems, and so now I'll introduce how we think male heterogametic systems may arise in sex chromosomes. So we'd have a sex-determining region on mammalian sex chromosomes. It was a testis-determining factor, and now it's called SRY, sex-determining region of the Y. And initially, individuals who inherit this allele, it's not a gene yet, it's just an allele. But it's an allele because it can swap back and forth between the 2 chromosomes. So individuals who inherit this allele will develop testes and make sperm, and individuals who do not inherit this allele will develop ovaries and make eggs. And for a while, you're not going to have sex chromosomes because it's just an allele. It's not a separate gene. There's nothing suppressing recombination between the chromosomes. But if there's a second allele, something that's sexually antagonistic, and in this case, we think it's something that would be beneficial for males and detrimental for females. That's the antagonism there. If there's something that could link the thing that's beneficial for males with the thing that determines maleness, then it would be selected for in the population. And our hypothesis is that it would be an inversion. So we see inversions routinely in the evolution of sex chromosome systems. And so we'll have an inversion, and now we can start to have separate sex-linked genes because there's no recombination going on. You can get neofunctionalization, specialization. They're not alleles, they're not swapping DNA anymore. But that's also a problem, right? Because in the absence of recombination, so we'll hear a little later, just the evolution of sex itself, having recombination is so beneficial, and we've seen it evolve and be maintained for millions of years. And now we have sex chromosomes and we stop recombining. And so there's consequences of that you cannot fix errors that occur in the absence of recombination. You have to get rid of the whole chromosome or the whole region. So we think there's more than one inversion, right? So we'll think about this. We have our ancestral autosomal pair, sex-determining allele, sexually antagonistic allele that becomes linked in recombination, suppressing recombination. So we'll have an inversion, and we'll have a second inversion. And because differences will accumulate in the absence of recombination, we actually expect in species where males recombine, and you have XY sex determination, you'll have different regions where some regions will be more differentiated from each other on the X and the Y chromosome, and some will be more similar to each other on the X and the Y chromosome. And we can actually observe this. So in 1999, Bruce Lawn and David Page reported these differences. So I shouldn't walk over there because they're recording it. So this group is synonymous differences. So we think largely neutral-ish differences accumulated between the X and the Y pair. So there's a group with a lot of differences between the X and the Y pair, a group with many differences between the X and the Y pair, a group with a few differences, and a group with not very many differences between them. So they proposed that there were 4 large-scale recombination suppression events, and they called them evolutionary strata, kind of referring to geologic strata over time. So there are these time-scale differences between the X and the Y chromosome. Only we'll swap it so that we have the youngest to the oldest, if we think about the X chromosome here with the short arm and the long arm of the X chromosome. In 2005, when we sequenced the human X chromosome, we could actually look for inversion differences. So we had the sequence of the human X chromosome, the sequence of the human Y chromosome. We could look at gene order and find a 5th inversion, so a 5th evolutionary strata was identified. One thing I'll point out is I'll show you the X chromosome on the next slide here. And these few genes, these 3 genes span almost 100 megabases on the X chromosome. And so some work that I did when I was at Berkeley with some people at University of North Texas, Rajiv Assad and Ravi Pandey, we developed a method to look for sequence similarity in regions across the X chromosome, and we showed that we could recapitulate some of the previous evolutionary strata boundaries, and we applied it to the whole X chromosome and inferred that there had been at least 9 inversion events, or at least 9 recombination suppression events based on sequence similarity. And all of these were previously defined to be that first old evolutionary strata. And it's not that what was done before was wrong, it's that synonymous substitution rate saturates over time. And so at a given point, you're just not able to tell if there's been a ton of difference or a really ton of differences, right? And so this method, we were able to kind of subdivide that a little more, even with our really old sex chromosome system. So then there's these 2 regions that still persist and still can recombine on the human sex chromosomes, and they're pseudoautosomal regions. So I guess this is the nice thing now about being an advisor is I get to embarrass people in my lab and put their faces up here all the time. So what we have recently studied, and we published this year, is looking at genetic diversity across the 2 regions that still recombine. So Danny and Sarah are here. They're actually both presenting new work, but you can ask them about this work, where we found that genetic diversity suggests that there's not a strict boundary. Even though we know there's an inversion that should suppress recombination between X and Y, genetic diversity doesn't suggest that that boundary is as strict as the inversion would suggest. And so we do know that things can get a little wobbly around inversion breakpoints. And so it has implications for when we're studying genetic diversity on the X chromosome and the autosomes, for which regions we say are truly X-linked and should have reduced diversity. Okay, so we know human X and Y chromosomes have differentiated from each other quite significantly. We know there are these tiny regions that still recombine. What about the rest of the chromosome? So if you have huge differences in chromosome content, and we know that aneuploidies tend to have severe phenotypes, how does it work out that half the population functionally has 1 X chromosome and the other has 2? And there's not, as far as I can tell, although we have some aging differences, really severe phenotypic differences between genetic males and females. And what happens is we have the evolution of X-inactivation. And so this is work done a little bit ago, and then I'll tell you about new stuff that we're working on. And so what happens in mammalian cells, as far as we can tell, is one of the 2 X chromosomes in individuals with 2 X chromosomes is silenced. And it forms a ball of heterochromatin that exists, as we were thinking and talking about earlier, kind of around the periphery of the nucleus. And so if you stain DNA, you can see in XX individuals this ball of heterochromatin that's one of the X chromosomes that's silenced. And in humans, it's actually a counting mechanism, or mammals as far as we can tell, eutherian mammals. So individuals with two X chromosomes will have one bar body, one of these balls of heterochromatin. Individuals with an X and a Y will not have one. And then if you have variations on that, so individuals with three X chromosomes will have two bar bodies. Turner syndrome, individuals with a single X will not have a bar body. And XXY, so individuals who develop testes but have two X chromosomes and a Y will have two bar bodies, or one bar body there, because they have one extra X chromosome. But there's still a phenotype to having different numbers of X chromosomes. So it suggests that that X-inactivation is not complete, and we actually know this. And so Laura Carroll and Hunt Willard in 2005 looked at 9 cell lines and identified, well, you'll see, right? So there's going to be some genes that escape that inactivation and some genes that are subject to the inactivation. There was recently just a bioRxiv preprint from Daniel MacArthur's group that looked across the GTEx data and actually recapitulated a lot of this and got a little finer scale resolution. So I encourage you to look at that publication. So what Carroll and Willard found, though, was they looked at one individual and they found that approximately 15 to 20% of the genes on that X chromosome escaped inactivation. But if they looked across 9 cell lines, they found that there were some regions that were routinely silenced in all individuals, some regions that were routinely escaping inactivation, and about 15% of the genes escaped heterogeneously. So they were not always silenced or always escaping if you looked across humans. And this has been recapitulated in the recent bioRxiv preprint. So our question was, how did X-inactivation evolve? So we clearly don't have a whole chromosome that's silent. There's some gene by gene mechanism going on here. And so what we did is we first asked, what's ancestral on the X and Y chromosome? We know there's been gene movements onto and off of chromosomes, and so we looked at the X chromosomes and compared using comparative genomics, human, mouse, dog, cow. We also used chicken as an outgroup and looked for genes that were conserved across these X chromosomes or the homologous region in chicken and identified a set of genes that we thought, these are likely what was on the ancestral X chromosome. And then for each of those, we did extensive sequence similarity searching on the human Y chromosome, and we knew genes that are functional on the Y chromosome, and we identified pseudogenes, so genes that have a single exon, a few exons, but are not functional, don't have a conserved open reading frame, or just pieces of genes on the Y chromosome. And then we had a set of genes that we could, no matter how generous we were being with our searching algorithm, we could find no evidence of them still persisting on the human Y chromosome. And we make an assumption here. And our assumption is that X-linked genes with a lost homolog, something that we can't find anywhere, were lost longer ago than those that still retain some pseudogene. So we think it takes some time. Certainly there can be large-scale deletions, and we have insertions and deletions. But largely we think the group that has a pseudogenized Y copy, that pseudogenization occurred earlier than those that have been completely lost from the Y chromosome. And so with that, we can now break up the X chromosome into those that have a functional Y copy, a pseudogenized, or a lost Y copy, and go back to that X inactivation profile and say, can we predict X inactivation status if we know about the Y-linked gene status? And we did linear discriminant analysis, and actually we do pretty well. We do about 90% accuracy predicting this, and I'll show you just in this cartoon form. So for every gene so here's genetic males, for every gene in an individual with 2 X chromosomes, if the gene escapes, or if the gene has a functional copy on the Y, then it escapes inactivation. There's maybe one example in the pseudoautosomal region 2, but we have to look at that, and I'll tell you why we might be suspect of it in a minute. For genes that have a pseudogenized Y copy, so these are ones that we think were more recently lost on the Y chromosome, those tend to be the ones that are heterogeneously silenced. So more recently lost the Y copy, you're silenced in some individuals and not in others. And largely, by and large, for genes that we cannot find any evidence of a Y-linked allele, they are inactivated or subject to silencing on the inactive X chromosome. And so it leads to this model. It's going to go off in 6 seconds. Cancel. Great. And so what we can think about is that initially you'll have equal expression from the X chromosome and equal expression on the X and the Y chromosome. And then as you start to lose functional gene content, we think gene expression going down. If dosage is important, so this is a caveat, for genes where dosage is really important, then you'll have compensatory mutations because we can't fix them on the Y chromosome in the absence of recombination. We'll have compensatory mutations, and we'll get upregulation of the X in males. Every X in a genetic male will be passed on to his genetic daughters, and so we'll get subsequent upregulation on the X in genetic females, and this process will continue, and it will get to a point where if it's dosage sensitive, suddenly now we have too much expression, and so there'll be some selection to silence one of those two copies. And so we, at least in this case, support that X-inactivation evolved in eutherian mammals gene by gene in response to gene loss on the Y chromosome. And so, right, we're here in the Center for Evolution of Medicine now, and so there's a lot of clinical relevance to studying X-inactivation. So in mouse models of Fragile X syndrome, so Fragile X syndrome is where there's a microsatellite repeat, and as the mice age, or any individual with Fragile X syndrome ages, the microsatellite repeat will increase, and you can lose a piece of the X chromosome, and it leads to some neurodegeneration phenotypes. In the mouse models, because X-inactivation is random, we can randomly silence the disease copy and have no phenotype in a mouse pup, or you can randomly silence the healthy copy and show the disease phenotype for individuals that are heterozygous here. So it'd be nice to know if we could figure out why it's random and if X-inactivation is really random. We'd also like to know, for GWAS studies, does your gene escape inactivation? Do you expect it to be heterozygous and have expression from both copies, or do we think that it might be subject to silencing? So there might be some evolutionary pressures acting on it. And we also know that X-inactivation is heterogeneous within tissues. So we don't have great evidence of this yet in humans. We have really visible evidence in calico cats with orange and black patterns, where the orange is one X chromosome, the black is the other. And depending on which X is inactivated, you'll get an orange or a black patch of fur. Jeremy Nathan's group did some good work in mice. And apologies, because red-green color blindness is X-linked, and we tag everything with red and green fluorescent tags. But I will tell you, for those that may not be able to see the difference, in the corneal endothelium, it's like I took a bag of red and green jelly beans and shook it up. So it's red, green, red, green. So we see very late that the X is inactivated in the corneal endothelium. But in the mouse model here, in the tongue, it's bilateral. So half of the tongue is one X and half of the tongue is another X. And the thing that is kind of got this really exciting response is that in the mouse brain, which is a little difficult to see here, you see a lot of patchiness here, but largely one hemisphere is one X chromosome and one hemisphere is another X chromosome. So we don't know what this is like in humans, but we are looking at the GTEx data and trying to say if we can infer when the X is silenced based on the shared tissue organization. So we know when different tissues develop, and we can try to infer based on which allele is inactivated in each tissue, when the X was silenced in humans. We're working with some placenta data, so I'm just going to shout out to some of the posters we have here also. So we're initially reanalyzing some data and really highlighting the importance of data quality. We're going to be getting some new samples from different human populations in the placenta to look at sex-biased expression and allele expression there in this tissue that's routinely expelled, but really critical for development. I'll talk later about the green anole, this handsome critter, where we're looking at dosage compensation and actually find strong evidence that there is also dosage compensation in this XY individual. I'm going to rush through because nobody's telling me to stop. I should be telling myself to stop. But you should also check out, we're working on a new method. Both Tim and Tanya are here, actually. Sorry. Sean is here. Tim is here. Kimberly is here. Valeria is here. Great. One of the challenges of next-generation sequencing is that because of shared homology between the X and the Y chromosome, if you align your next-gen sequence from an individual without a Y chromosome to a reference with a Y chromosome, some of those reads will misalign to the Y chromosome, and you will both undercall variation, you'll have reduced power to call variants there, and you'll undercall, I anticipate, we're looking at it right now, gene expression, because you'll have reads misaligning between the X and the Y chromosome, especially for pseudoautosomal regions. And so we're working on a method to try to fix that a bit. Right, and so Thank you all very much.